Due to environmental pollution and increased stress, our immune system is becoming unstable, and combined with changes in diet, the number of allergy sufferers is rapidly increasing. For individuals with food allergies, even trace amounts of allergenic substances can pose life-threatening risks, making it crucial to accurately identify allergenic ingredients in processed foods. To address this issue, BIONEER offers the AccuPower Allergen qPCR product line, providing a solution that enables precise and effective analysis of food allergenic substances. Real-time PCR using probes employs fluorescent probes, approximately 20 base pairs in length. The probe contains a reporter dye at the 5-foot end that emits fluorescence and a quencher at the 3 end that suppresses this fluorescence emission. During synthesis, the template DNA denatures from double-stranded to single-stranded, and the probe binds to a specific complementary sequence on the single strand. As Taq polymerase synthesizes the complementary strand, it degrades the probe sequence, causing the reporter dye to separate from the quencher and emit fluorescence. The emitted fluorescence is measured in real time to determine the presence or absence of the specific gene. The AccuPower Allergen Real-Time PCR Kit contains all the essential components required for PCR reactions, enabling reduced preparation time and designed for ease of use. By applying hot start Taq DNA polymerase technology, nonspecific reactions are reduced, thereby increasing the product sensitivity. Additionally, it amplifies specific genes to identify various allergenic ingredients and can accurately detect low DNA concentrations down to approximately 20 copies per test. BIONEER offers 27 different kits for detecting allergenic substances, providing optimized tools for food safety management. AccuPower Allergen Real-Time PCR Kit.